In this video, we'll be covering different types of receptors. Receptors are a specific kind of membrane protein responsible for receiving a signal from outside of the cell and transmitting it into a signal inside the cell. This kind of communication between cells is critically important because cells within the same organ system and in different organ systems need to be able to coordinate their responses to various stimuli. The first kind of receptor we'll discuss is the ligand-gated channel, otherwise known as a channel-linked receptor. This kind of receptor has the machinery to both receive a signal and allow passive transport of ions all in one protein, so it is both a receptor and a channel. The way these proteins work is the receptor part of the protein first receives a signal from the external environment. So you can see that here, where this orange signaling molecule is binding to the receptor binding site. The channel part of the protein is only open once the signaling molecules are bound to the receptor, which allows for the regulated flow of ions into the cell. For example, when two molecules of acetylcholine bind to a nicotinic receptor, the nicotinic receptor channel opens and allows the flow of positive ions into the cell. This process is important for skeletal muscle contraction. If these channels weren't gated and were just open all the time, then our skeletal muscles would be contracting all the time, which would be bad. So being able to regulate these channels with a ligand is really important. The next kind of receptor we'll talk about is the enzyme-linked receptor. These receptors are both receptors and enzymes all in one protein. The enzyme portion of the protein is only active when a ligand is bound to the receptor portion. Often the enzyme that's being activated by the receptor binding is a tyrosine kinase, which phosphorylates tyrosine residues on proteins. This acts as a sort of on-off switch for many cellular functions. An important aspect of receptor physiology is that many receptors have one primary effect, so in this case it's going to be the activation of the tyrosine kinase enzyme, but they also initiate a cascade of many downstream effects, and the molecules involved in these downstream effects are called secondary messengers. One of the secondary messengers that is activated by receptor tyrosine kinases is phosphoninositide 3 kinase, or PI3K. PI3K goes on to activate the signaling molecule AKT, which goes on to activate mTOR. The PI3K AKT mTOR pathway is important in promoting cell growth, differentiation, and survival. Another signaling cascade activated by receptor tyrosine kinases is the Ras-RAF mitogen-activated protein kinase pathway. These proteins are involved in regulating the transcription of genes involved in the progression of the cell cycle. Tightly regulating these processes is extremely important because if these proteins were always active, you would have a constant proliferation of cells. And that's what leads to cancer. In fact, the Ras protein is an important oncogene. A mutation leading to the overactivation of the Ras protein is a common molecular cause of many cancers. Examples of receptor tyrosine kinases include the insulin receptor and epidermal growth factor receptor. Notice that these are both receptors that signal to the cell that there are nutrients available in the environment and it is time to grow. The next class of receptors we'll talk about are intracellular receptors, which rather than being on the membrane surface are found inside the cell. This means that their ligand needs to be membrane permeable in order to pass through the membrane and reach the receptor. An example of a membrane-permeable ligand is nitric oxide, which passes through the membrane to bind to soluble guanylate cyclase. This leads to the activation of cyclic GMP and protein kinase G. This pathway is important for processes involved in vasodilation, and a variety of drugs that are used to promote vasodilation, like the angina drug nitroglycerin and the erectile dysfunction drug sildenafil, act by increasing signaling in this pathway. Another important class of intracellular receptors are nuclear receptors, which are not just inside the cell, but in the nucleus. These receptors are directly involved in regulating gene transcription, which has a much more long-term effect on cell function compared to the transient increases in secondary signaling molecule activity induced by other kinds of receptors. In fact, most nuclear receptors are activated by hormones that are intended to have a large global effect on the body and the cell, such as steroid hormones and thyroid hormones. The last kind of receptor we'll talk about are G-protein coupled receptors. These receptors are membrane proteins with 7 transmembrane domains. On the outside of the cell, the receptor binds to a ligand, and then on the inside of the cell, it activates its coupled G protein. G protein coupled receptors are the most common pharmaceutical target and include receptors like the adrenergic receptors, opioid receptors, dopamine receptors, and many more. The G proteins that couple to this receptor are what induce the downstream signaling effects in the cell. G proteins are heterotrimeric proteins, meaning they're made up of 3 different subunits: the alpha subunit, the beta subunit, and the gamma subunit. When the G protein becomes activated following ligand binding to its G protein coupled receptor, the G alpha subunit separates from the G beta and G gamma subunits. The kind of signaling that is induced within the cell depends on the subtype of the G alpha protein. G alpha S proteins are called that way because they stimulate the production of cyclic AMP, which leads to the activation of protein kinase A. An example of a GS protein coupled receptor are beta-1 adrenergic receptors. GI proteins are called GI because they inhibit the production of cyclic AMP, which leads to less activation of protein kinase A. An example of GI protein coupled receptors are the opioid receptors. And then the last common type of G alpha subunit we'll discuss are the GQ alpha subunits. And these act on a different pathway than the cyclic AMP and pKa pathway. GQ proteins activate phospholipase C, which cleaves a molecule called PIP2 into IP3 and diacylglycerol. IP3 causes calcium to be released from the endoplasmic reticulum, and diacylglycerol activates protein kinase C. Muscarinic 1 receptors are an example of a GQ coupled receptor. I recommend practicing drawing out these signaling pathways so that you know which G alpha subunit pairs with which downstream signaling molecules. And we'll continue to discuss G protein coupled receptors throughout the pharmacology series. So to sum up, receptors are proteins responsible for receiving a signal from outside the cell and turning it into a cellular response. Ligand-gated channels or channel-linked receptors are proteins that receive a signal from a signaling molecule and open a channel to facilitate the passive transport of molecules and ions. Receptor tyrosine kinases are enzymes that receive a signal and then phosphorylate tyrosine residues, leading to downstream activation of PI3K, AKT, and mTOR, as well as RAS, RAF, and MAPK. Intracellular receptors are found inside the cell, and nuclear receptors are found inside the nucleus, where they regulate gene transcription. Finally, G protein coupled receptors are transmembrane proteins that receive an extracellular signal and activate an intracellular G protein. This G protein activation can lead to either activation or inhibition of the cyclic AMP pKa pathway, or it can lead to phospholipase C activation depending on the G alpha subunit type. Because receptors drive so many cellular functions and have evolved to bind small molecules, they make excellent drug targets.